Bom dia, estamos em mais uma sessão, setembro de 2025, mais uma sessão do nosso SIG Farmaco. É com prazer que eu, Maria Cristina Avelar, aqui da UFCSPA, Escola Paulista de Medicina, estou abrindo aqui na coordenação desse grupo, essa sessão que é o Special Interest Group em Farmacologia Terapêutica, uma plataforma de encontros mensais desse nosso grupo que chamamos o SIG Farmaco. No próximo slide, professora Helena, por favor. O SIG Farmaco está inserido na rede universitária de telemedicina, que é uma iniciativa do Ministério da Ciência e Tecnologia e Inovação, o MCTI, apoiada pela financiadora de estudos e projetos FINEP, pela Associação Brasileira de Hospitais Universitários e coordenada pela Rede Nacional de Ensino e Pesquisa, ARNP. O nosso SIG Farmaco, ele tem como objetivo uma estrutura de vídeos e webconferências. Então, no próximo slide, do sistema RUT da Rede Universitária de Telemedicina, e o grande objetivo aqui é, de fato, reduzir as distâncias geográficas e estimular a integração e colaboração em ações de ensino e pesquisa a todos os interessados na área de farmacologia e terapêutica no Brasil. No próximo slide, estamos no nosso 7º ano de atividades. Nesse período, nós tivemos várias instituições, como vocês podem ver no slide, que estão aqui integradas, né, nessas nossas atividades, e os participantes, eles em geral são estudantes, pesquisadores, professores e profissionais da saúde, incluindo profissionais em hospitais universitários, que têm interesse em ensino, pesquisa e atividades educacionais na área, né, e temáticas de farmacologia. Eu sou, então, a Maria Cristina Avelar, uma dos integrantes do grupo coordenador desta atividade. Então, comigo estão o professor Emiliano Barreto, de Alagoas, a professora Roseli Godinho, aqui também da UFCSPA, Escola Paulista de Medicina, o professor François Noel, da UFRJ, e o professor Paulo Guedini, da UFG, da Universidade Federal de Goiânia. E essas atividades, elas têm sido construídas ao longo dos anos pelas nossas coordenações e em apoio da rede RNP e da Sociedade Brasileira de Farmacologia e Terapêutica Experimental. No próximo slide, nós lembramos que durante essa sessão, nós pedimos que todos mantenham seus microfones desligados. Lembramos que existe um ícone CC que dá o closed captioning, então os participantes têm a possibilidade de traduzir a apresentação para as línguas de inglês, espanhol ou manter em português. Durante a sessão, a qualquer momento, vocês podem estar encaminhando perguntas, comentários pelo chat, e lembrar que existe um registro, né, de presença na sessão, e que no próximo diapositivo, Helena, por favor, todos que entraram aqui fizeram o seu registro pelo link, né, que foi disponibilizado no folder da sessão. Todos que fizeram isso, normalmente esse registro de presença já é automaticamente realizado. No próximo diapositivo, lembrem-se apenas que durante o seu registro e durante o login aqui para essa área, que vocês tenham indicado, né, então o desejo registrar presença nessa sessão. Então, isso é válido durante o período da sessão, que é das 9 até as 10h30 da manhã, e depois, posteriormente, cada participante tem direito a fazer o download do seu certificado aqui de participação. No próximo diapositivo, vou dar brevemente aqui a estrutura da sessão. Então, aqui os primeiros slides, né, para fazer as boas-vindas e falar um pouco dessa nossa atividade. A seguir, eu passarei a palavra para a professora Helena Barros, que está como moderadora desta sessão e que foi a que apontou e indicou o nome do professor palestrante hoje desta sessão. Então, ela fará uma apresentação do professor. A seguir, teremos o palestrante apresentando os seus slides e a apresentação nos próximos 30, 40 minutos. E a seguir nós temos um período que é para o debate, para as discussões, para os comentários, lembrando a todos, né, que então a qualquer momento contribuam encaminhando as questões via chat. E ao final eu voltarei aqui para fazer o encerramento e anunciar então o próximo Sig Farmaco de outubro. Próximo slide. Então, em nome do grupo Sig Farmaco, já agradecendo à professora Helena Barros pela sugestão do palestrante, agradecendo ao professor Graziano Pinna por estar aqui. So I'll change to English because just to tell you, Professor Pinna, thank you so much for accepting our invitation. And I'm also thankful to Professor Helena Barros for being here with us. And so for now, I'll leave the session and the floor is yours. Thank you so much.
Thank you, Professor Maria Cristina Avelar, for this nice introduction. I'm very, very glad that we are here meeting this morning with Professor Graziano Pinna and all the audience that we have. And also thank you, Professor Graziano, for being with us now for so many years with such a prolific production and training of our students here in Brazil. I'm going to change to Portuguese because Professor Pinna has been studying Portuguese and he can understand everything that we speak in Portuguese also. So to introduce Professor Pinna, I want to say that o professor Graziano Pinna é professor no Departamento de Psiquiatria da Universidade de Illinois, em Chicago, e ele também é mentor de programas interdisciplinares de pesquisa a respeito de saúde da mulher, que é uma área que tem se desenvolvido muito nos últimos anos. Ele tem um doutorado em neurofarmacologia, neurociência na Itália, e ele tem trabalhado nos Estados Unidos, na Universidade de Illinois, em Chicago, em torno desde 2011. Tem um trabalho extremamente prolífico, com muitas atividades de ciência, de pesquisa e de inovação, trabalhando principalmente com as desordens psiquiátricas relacionadas a estresse, com PTSD, com ansiedade, com depressão, então, que é o mote, digamos, da nossa fala de hoje, ou da fala dele de hoje, e basicamente trabalhando no impacto da biossíntese de neurosteroides no sistema nervoso central, especialmente a alloprenolona, do ponto de vista de regulação emocional e do comportamento. Ele tem vários trabalhos translacionais, utilizando modelos animais e pesquisa humana para trabalhar e desenvolver as teorias que ele vai parcialmente nos apresentar hoje. Ele trabalha com vários sistemas de neurosteroide integrados com o sistema endocannabinoide, com o sistema de, eventualmente, abuso de substâncias, além das manifestações de estresse e de depressão, sempre associando as diferenças de gênero para a apresentação desses diferentes efeitos. Ele publicou muitos artigos de alto impacto e, basicamente, tem contribuído para a compreensão dos mecanismos que têm levado a avanços terapêuticos, inclusive com o uso da brexanolona como um tratamento para depressão puerperal. Então, quero com isso agradecer mais uma vez essa parceria de hoje e de muitos anos, que tem sido continuada, não só aqui em Porto Alegre, na Universidade Federal de Ciências da Saúde, mas também em várias outras universidades do Brasil. Professor Graziano, muito obrigada mais uma vez, e o microfone é seu.
Thank you very much, Helena and Christina, for that very kind introduction. And it's always a pleasure to be here and have an opportunity to show the results of my work in the Department of Psychiatry in Chicago. So today I will speak about neurosteroids, as Helena has mentioned, and the role that these neurosteroids have had in the past decades, mainly in shaping our mind, and how this is a promise for the future, and how it will shape psychiatry and precision medicine of mood disorders, but in general of all neuropsychiatric disorders. And in fact, these molecules are involved play a role in the pathophysiology of many disease and disorder that goes from neurology to psychiatry. I will try to cover a little bit of this and also having an historical approach on how these neurosteroids were discovered and later on were approved for the treatment of neuropsychiatric disorders. But before doing so, I will start with my disclosures. In fact, I was a paid or unpaid consultant and advisory board member for a number of companies, and also the University of Illinois at Chicago has submitted 2 patent applications on my behalf that were allowed in the past 2 years. And also my work was supported by several funding agencies in the United States, such as NIH, NIAAA for alcohol use disorders, Department of Defense, and Veteran Affairs, and more recently, Michael Rees, a pioneer in research award. So neurosteroid and how, next slide please, how my journey started in this field has covered the past almost 2 decades and 25 years. And in the beginning as a student in University of Cagliari, I had no idea that this work would develop in a couple of decades in the way it did. And what started with a curiosity as a student became then a scientific passion and mission, and I shaped my career as a young scientist. I still continue to shape my career today. This journey was not a straight line. Like many discoveries in science, it began with unexpected observation, and I didn't imagine that this would become such a central focus of my career, leading to translation advances and collaboration with industry and with several other universities in the United States, and also here in Brazil. And today I want to share not only the science, but also the story of how this field grew and how still is growing and is affecting the life of many people and patients we need around the world. Next slide. This work I was influenced by pioneers in the field of neurosteroids, and I had the opportunity to learn from Dr. Costa, for example, with whom I worked for about 10 years. It was a very detail-oriented scientist, and he has affected neuroscience and neuropharmacology for over 60 years, publishing more than 1,000 papers. He was also a member of the National Academy of Science of the United States, and working together with Alessandro Guidotti. Next slide. They shaped the field of neurosteroid. I'm really thankful and grateful for the contagious patient that they gave me in this line of research of neurosteroid and neuropsychiatric disorders. So this scientist have contributed really a lot in neurosteroids. And neurosteroids, talking about mainly of allopregnanolone, was discovered in about 80 years ago, in 1938. And during this time of 80 years, numerous contribution in the field, including the discovery and understanding of the fast and non-genomic mechanism of this molecule and of allopregnanolone and other GABAergic neurosteroids that have the ability and as a mechanism of action, the potentiation of membrane neurotransmitters such as GABA receptor, or the ability to affect our neurotransmitter systems such as glutamate and NMDA receptor that I will discuss during the course of my presentation today. From this discovery of acting in this neurotransmitter receptor to the ability of affecting behavior by inducing anxiolytic, antidepressant effect in both animal models and in humans, to the design of first clinical trials that have led to the discovery that allopregnanolone has the ability of induce very fast antidepressant effects, and that led to the approval of allopregnanolone first as an IV formulation in a compound called brexanolone for the treatment of postpartum depression. This was approved by the FDA in 2019. And then a synthetic analog of allopregnanolone with the ability of being administered orally has shown efficacy in both postpartum depression and some mixed effect on major depression. And so for this efficacy in treating postpartum depression, this oral formulation was approved by the FDA in 2023. So I will cover a lot of these discoveries in the field of neurosteroid research, and I will talk also of the approach of my lab. My lab has applied the translational research approach to study neurosteroid, learning from neurobiological deficits of biomarker candidates quantified in subjects that meet criteria for PTSD and depression and postpartum depression, developing animal models for this condition, including the social isolated stress mouse model of PTSD and depression, and more recently also working on animal model of alcohol use disorder and testing new molecules in this model. Followed by the translation of finding into the clinical setting with the goal of developing a personalized precision medicine for neuropsychiatric disorders. So ours and many other significant studies, clinical and basic research studies, as we see in this next slide, in various teams that have worked, Helena, next, okay, thank you, that have worked in the field of PTSD and depression, have contributed to create a translational model for this condition. In my lab, we have used social isolation stress, and we found that social isolation, and we probably can recall how we were feeling during the pandemic, the COVID pandemic, when a lot of us were isolated as well, and how we felt during that condition. Social isolation is a very potent chronic stress form that induces a lot of changes neurobiologically and behaviorally. Neurobiological, in rodents, we have found that social isolated animals express deficit in fear extinction, anxiety, and depression-like behavior, and also very high level of impulsivity and aggression, which is really very interesting because this reproduces suicide behavioral traits in humans. And this deficient behavior are associated with deficient allopregnanolone biosynthesis, lack of pharmacological effect of benzodiazepine, and changes in GABA receptor subunits, and also changes in deficient production of neurotrophic factor. So as you know, all these aberrant neurobiological factors are also been observed in humans. And the work that we have done in my lab is always comparing animal model with the observation that we have done in humans, and then go back to the animal model, test new treatments, and go back to the humans and develop clinical trials to study the efficacy of new molecules with the goal of improving precision medicine for this condition. Next slide, we see that a critical step in clinical research is the ability to find the right patient and successfully enroll them in clinical trial. Recruitment ensures that patients meet the specific inclusion or exclusion criteria, and this improves the reliability of the study. Trials progress efficiently, avoiding costly delays, and researchers capture the right population to evaluate the safety, efficacy, and generalizability of a new intervention. Effective patient recruitment is, though, essential for bridging discovery with the clinical application, and so ultimately bridging new treatments to the person that are in need. As you know, right now, all treatment in psychiatry are all discovered either by chance or there has been some development to try to follow some mechanism of these drugs. But as you know, there are no biomarkers that have bine established in psychiatry and neuro psychiatry in general. And so, for example, SSRI, such as fluoxetine, sertraline, Paxil, are all molecules that are given as a fit, one fit for all, without any guide and any information that we can have from the patient in terms of biomarkers. And so this is also a big problem because we know that this molecule, 50% of the patients do not respond to SSRI, and probably because a lot of patients that develop depression, PTSD, and other neuropsychiatric disorder, they do not have a deficit of serotonin. Maybe they have other deficits that are not covered by SSRI. And for this, it's really very important to develop new molecules that target specific neurobiological and deficit in this patient. So this is what we would hope to see, and next slide, in a development of translational research, which is a stepwise process, and it's a continuum process that is often described across 5 stages, from T0 to T4, T0 with scientific discovery, laboratory-based and preclinical studies that generate fundamental insight and identify potential therapeutic targets, followed by a translation to humans. So the first in human or phase 1 clinical trial, there is the goal to assess safety, dosage, and feasibility of a given drug. Phase 2 and phase 3 clinical trial are then designed to evaluate efficacy, refine treatment protocol, confirm safety in a larger patient population. And so this is really very important because phase 2 and phase 3 clinical trials are the most important because upon a positive effect in this trial, this is usually followed by the approval of a given drug for treatment of a specific disorder. Phase 4 is the post-marketing and post-approval trial that assess real-world effectiveness, safety, and long-term outcomes. So this framework ensures that discovery follow a rigorous path, bridging the gap from the bench to the bedside, to population health, while continuously generating a feedback to refine intervention. As you know, next slide, it takes about 15 years to discover one molecule out of 1,000 that has been studied and tried to be developed. So while thankful not only to have witnessed something like allopregnanolone approval as a drug for mood disorder, given that my research was involved into the process of understanding mechanism and behavioral action and pharmacological implication of allopregnanolone in mood disorder and PTSD and alcohol use disorder. At the same time, I also understand that there is so much work that needs to be done to improve patients' life. And also to discover new molecules that are neurosteroid-based compound and that can be developed in a way by developing and discovering biomarkers so to improve the efficacy of this drug and help a much bigger proportion of patients in need. So in the next slide, we see that this work is really very important for our patients and for people in need. However, there is like a huge commitment, and I'm saying this mainly for young investigators that are listening and they're part of the Brazilian Pharmacology Society to inspire them, but also to make them aware that this work really is a commitment in time, energy, patient, and it's not a straight line, and it's a really a path that is made by daily gains, and it's a step-by-step process. It's very important to have a lot of determination, patience, motivation, curiosity, resilience. There will be many moments in which we fail and we are discouraged, but it's also really very important to understand that big gains are made also with moment of failure. And what is really very important to progress each of careers is also important to collaborate, share, generosity, training, and keep the vision alive. So these are all very important characteristics to be a successful scientist. And as I mentioned, this is a message mainly for all the youngest individuals that are following this presentation. So as I mentioned, the focus of my research for the past 2 decades has been mainly on neurosteroid and the progesterone derivative allopregnanolone, and I want to focus a little bit on this and also to explain what these molecules are and where are produced and what do they do. And for example, allopregnanolone is a potent positive allosteric modulator of GABA receptors and acts as an endogenous tranquilizer. Helena, go on with the slide. And has also an antidepressant function. But we also study the sulfated forms of these neurosteroids that have an effect on inhibiting excitatory neurotransmission, which is mediated by NMDA receptors. So this is really very interesting because it gives, we have on one end, one mechanism, there is potentiation of inhibition, and on the other end, we have inhibition of excitation. So this mechanism is really very important to keep the fine balance of excitatory inhibitory neurotransmission, which has a very important role in maintaining a proper behavior, and also regulating and improving behavioral dysfunction. So neurosteroids, it's a discovery of the past 30 years. Allopregnanolone is not only produced in adrenal glands and gonads, as we have always known from a long time, but Beaulieu, in the French scientist, discovered that in the 80s, allopregnanolone can be also produced in the brain, and allopregnanolone, Helena continued with clicking. Allopregnanolone in the brain is produced in several cells and neurons, and our work, continue please, our work has shown that allopregnanolone and the enzymes that produce allopregnanolone are expressed in neurons, in pyramidal neurons, in glutamatergic neurons of the cortex, hippocampus, and pyramidal-like neurons of the basolateral amygdala. Mainly, this work that I'm showing here is the expression of alpha reductase, and this was the work of one of our PhD students, Roberto Carlos Alcis Balboa, who is a scientist now in Spain. So his work was really pivotal to show that these enzymes are expressed in pyramidal neurons, both in the mouse brain as well as in the Brodmann area 9 of the human prefrontal cortex. And as you see here in the picture showing the merge of 5-alpha reductase and vesicular glutamate transporter 2, which is a marker for glutamatergic neurons. This is a clear proof that allopregnanolone can be synthesized in neuron, given that 5-alpha reductase, which is one of the most important enzymes that produce neurosteroids, is expressed in these neurons. Next. So, and in these neurons, allopregnanolone plays a really very important neurophysiological role in activating GABA receptors, and mainly extrasynaptic GABA receptors that express the delta subunits, for which allopregnanolone has an increased affinity. And by this mechanism, allopregnanolone regulates emotional behavior. So one of the things that we did in my laboratory in Chicago was to develop a methodology that allows the quantification, next slide, of this neurosteroid by gas chromatography mass spectrometry. This is considered the state of the art of neurosteroid quantification. And we perform this quantification in saliva, serum, plasma, CSF, postmortem brain. We collaborate with a lot of universities and companies, mainly in the United States. And working with HCMS is highly innovative and powerful for biomarker discovery in neuropsychiatric diseases because of the high sensitivity and chemical structural selectivity. We studied their role as possible biomarker for depression, postpartum depression, PMS, PMDD, PTSD, and in alcohol use disorder, with a special emphasis on women mental health. And now, next slide, thank you. So why is important to use this methodology is because, for example, if we use ELISA or radioimmunoassays, so methodology that are based on antibodies, we just, first of all, we have a large cross-reactivity with a number of other steroids. And second, the antibody do not distinguish the stereoisomers. With a gas chromatography mass spectrometry, we are able, for example, to detect and selectively quantify all the 4 stereoisomers of allopregnanolone. And why is this important? It is really very important because 2 of these isomers are positive allosteric modulator GABA receptors, while other 2 here color-coded in red are negative allosteric modulators of the function of allopregnanolone on GABA receptor. Was substantially what the antagonists do, they block the positive function of allopregnanolone and pregnenolone on GABA receptors. So if we measure these compounds with an antibody, we would have just one value that summarizes all these isomers, but this has no value in increasing our understanding, functional understanding and mechanistic understanding of this molecule in a given disorder. So unfortunately, a lot of papers are still, I mean, not a lot, but there are some papers that are still appearing in the field of neurosteroids where people quantify, investigators quantify allopregnanolone by ELISA. And so this should not be done because it does not increase the knowledge of the field. I wanted to make a really clear point on this because this is really very important, and because this also gives you the tool of sometimes reading and understanding papers and also understanding the pitfall that are inherent to a given study. In the next slide, we show that using this methodology, we were the first, and this was the work of Alessandro Guidotti and Erminio Costa and Vesca Uzunova. They, for the first time, showed that allopregnanolone level, quantified together with the other positive stereoisomer pregnenolone, is decreased in the CSF of depressed patient. And so this decrease of allopregnanolone was inversely correlated with the severity of depressive symptoms. More recently, with the work of Roberto Carlos Achis-Balboa, we have also quantified the expression of 5-alpha reductase in the human prefrontal cortex, and we found that allopregnanolone biosynthesis was also decreased in these prefrontal cortical neurons with an absence of changes in the cerebellum. The work of Vesca Uzunova in the next slide was really very important, and it was a great inspiration for a number of other studies that followed. So it was a really seminal discovery because she measured allopregnanolone before, as you see here in this blue square, we see that she measured allopregnanolone before and after treatment with SSRIs, including fluoxetine and fluvoxamine. And she observed that in the patient that improved the depressive scores, there was also an increase of allopregnanolone. So this, for the first time, showed that one of the mechanisms of SSRI could include a neurosteroidogenic effect. So this discovery later on, I translated it, next slide, in an animal model. So we have a back translation from the clinic into the basic research, and I created an animal model, as I mentioned previously, the socially isolated model. And we see that during social isolation of 8 weeks, there is a time-dependent increase of aggression, but also a time-dependent increase of depression-like behavior, anxiety-like behavior, and deficit in fear extinction. This is accompanied by a time-dependent decrease in allopregnanolone biosynthesis, including the expression of 5-alpha reductase, probably through epigenetic mechanism, and also a decrease in the level of allopregnanolone that was quantified in many corticolimbic areas, including the hippocampus, the prefrontal cortex, the basolateral amygdala. So all cortical limbic circuitry are involved in the appearance of this behavioral deficit. So what I did in several experiments that are summarized in this next slide, by using the stereoisomers of fluoxetine and norfluoxetine, I found that these molecules increase allopregnanolone in a stereoisomeric fashion, and the same concentration that increased allopregnanolone were also the concentration that were improving behavior, such as, for example, aggression. However, the concentration that were improving behavior were much lower than the concentration of this molecule, at least 10 times lower than the concentration that were affecting ex vivo serotonin reuptake. Also, these molecules, the stereoisomers of fluoxetine and norfluoxetine, fail to elicit a steroidogenic function on serotonin reuptake inhibition. So what does this research shows? It really provides a proof that SSRI mainly work through upregulation of neurosteroids rather than by inhibiting serotonin reuptake. And you know, after all, you may note in the literature that there is, as of today, no proof that SSRI really elicit antidepressant effects by inhibiting serotonin reuptake. There are many other mechanisms that have been proposed for these molecules, and my lab and also the research of Dr. Costa and Guidotti support a neurosteroidogenic effect of this molecule as the main mechanism for improvement of behavior. And that's why many years ago, we also renamed this class of compounds as selective brain steroidogenic stimulants to include under the same umbrella a number of molecules that share the same common mechanism of action, which is the neurosteroidogenesis and increase of allopregnanolone levels. So this discovery also is very important because shows that, next slides, that 10 times lower SSRI doses may be required to treat the mood disorder through a neurosteroidogenic mechanism. So less is more. You know, you probably know the architect Mies van der Rohe, a modernist, that introduced the concept of less is more in architecture. And this applies really very much also in psychiatry, because unfortunately, if a patient is going to a psychiatrist and the psychiatrist prescribes 20 milligrams of fluoxetine, fluoxetine doesn't work. At 20 milligrams, what do? They never think about giving 10. What they think is giving 40, doubling the dose of a given drug rather than giving a lower dose. And then increase to 80 milligrams and more side effects appear. And a lot of these side effects sometimes mask also the beneficial pharmacological effect of a drug. And so this is really a problem in psychiatry in general, where there is a large number of patients who do not respond to SSRIs, and most often psychiatrists think about giving more when they should actually give less to decrease side effects and to increase the efficacy of a compound. So after this discovery was done, although we didn't run any particular clinical trial using low doses of SSRI, however, in the clinical application, this discovery of the low doses of SSRI, excuse me, had an impact on the clinic. And in fact, in the next slides, we see that a lot of psychiatrists and clinical psychologists have observed that low doses of SSRI were really very important in protecting individuals from the hormonal withdrawal during premenstrual syndrome and PMDD. Also, low SSRI doses were also important to prevent or avoid suicide in children. And also low doses have been applied for the treatment of anxiety in children with autistic spectrum disorder. And in the next slide, we also see that there was a systematic review that has been published recently, a few years ago, in Lancet Psychiatry. And so this paper that included thousands of individuals that were treated with serotonin reuptake inhibitors shows that low doses offer an advantage in the treatment of major depression over large doses, and also these low SSRI doses were better tolerated with lower side effects. And next slide, thank you. And so this is really very important, because looking at as a whole in the population, it's really true that those low doses are much more important and much more efficient in treating psychiatric disorder rather than the large doses that induce a number of side effects. So finally, a year ago, we were able to receive this grant from the NIH to study low doses of sertraline in individuals with a premenstrual dysphoric disorder. And also we will look into the GABA receptor plasticity. What we will do, we will try to develop predictive biomarkers for these conditions. And we will not look, next slide, only to allopregnanolone biosynthesis, because one of the big questions of the past years was whether allopregnanolone as a whole is a marker for psychiatric disorders. And our research and research of other people also have more and more convinced us that we cannot look just into total level of allopregnanolone, but there is much more that we need to look into. And this is also include the enzymes that produce allopregnanolone and the receptors where allopregnanolone work. Next slide. Next. And in fact, some years ago, I came up with the idea that the biomarker axis would be the best to describe the neurobiological changes occurring in individuals that suffer neuropsychiatric disorder. And just because the literature is very mixed when people try just to predict changes in mood, for example, by just looking into total level of neurosteroid. There must be something more than that, much more complex. And so this biomarker axis, next slide, it will be really very important because it will contribute into the characterization of which this is a disorder, looking even not only at the neurosteroidogenesis, but also in the crosstalk between neurosteroid and other systems, such as the HPA axis, as well as the endocannabinoid system. A lot of connections have been discovered recently between the neurosteroid and endocannabinoid system. And this is really very important next, because as you know, there are a lot of comorbidities and there is a lot of overlap in the symptoms between mood disorders, for example, as there are a lot of comorbidities between depression, alcohol use disorder, anxiety spectrum disorder, And so a better characterization of our clinical population is really very important, and this cannot be only inferred by looking into total quantification of neurosteroid, but we have to construct a more elaborated approach, and this can come through the discovery of biomarker axis, and which will guide the personalized and precision medicine of neuropsychiatric disorder. Next slide. And so this is also the approach that we are taking in this newly awarded grant, where we will study neuroactive steroid biosynthesis, which includes hormonal levels or metabolomics, as well as studying the enzymes that produce allopregnanolone in the blood, which is collected by PAX gene tubes, and PAX gene tubes are made in such a way to preserve mRNA and DNA from this patient that can be extracted to studying then the genomic, through a genomic approach, GABA receptor subunits that are the receptor where neurosteroid act, and enzymes that produce allopregnanolone. And so we will do this in a study of postpartum depression. And why? Because postpartum depression is also the disease where neurosteroids were approved, brexanolone and zuranolone. Next slide. And pregnancy introduces profound physiological and hormonal changes, particularly a significant rise in estrogen and progesterone. Progesterone increases up to 100 times during pregnancy, and this may alter stress and trauma responses. So while these changes can provide a buffering effect in women that suffer from stress and trauma, in some of these women, these hormonal changes increases also vulnerability for anxiety, depression, and PTSD. Factors such as history of trauma, interpersonal stress, or lack of social support can increase this susceptibility. Stress during pregnancy has been linked to adverse outcomes, including perinatal depression, and potentially also predispose offsprings to future mental health disorders. So what we believe is that during pregnancy, both allopregnanolone and progesterone allopregnanolone increase, and then during delivery, as we know, there is an abrupt decrease of progesterone, but also allopregnanolone. We believe one of the factors can be that allopregnanolone decrease is much lower in individuals that go on to develop postpartum depression, as is shown in this study that we did in women that were exposed to trauma and that developed PTSD during pregnancy. So this hypothesis was tested recently in a study that I did in collaboration with Jennifer Payne and Lauren Osborne. Next slide. And we found that the ratio of pregnenolone, which is the positive allosteric modulator, to progesterone is decreased. So signifying lower production of pregnenolone in the second trimester and also the third trimester. So this is really important because this is an index or a biomarker that can give you some information on people that later on during the postpartum period can develop perinatal affective symptoms. Next slide. So another thing that we looked into was the relation between the positive allosteric modulator, so neurosteroid positive allosteric modulator, and that are color-coded here in blue, versus the antagonist isomers that are color-coded in red. Next. So we found that in this pregnancy study, the ratio of isoallopregnanolone to pregnenolone was increased, showing that the antagonist isomers were increased over the positive modulator. So this is really interesting because next slide, it shows that there is an increase of the antagonist and a decrease of the agonist. So this signifies that there is a change in the balance next in the positive versus the negative, and so a decrease of the inhibitory excitatory neurotransmission balance that could be one of the factors underlying the development of symptoms in mothers that develop postpartum depression. So in a few words, this could be a predictor for postpartum depression. This could be measured during the second or third trimester of pregnancy, and could be a red flag to expect a certain individual to develop some months later postpartum depression. This is very important because this matter could be monitored, could be treated in order to prevent the development of symptoms of postpartum depression or to worsen the symptoms of postpartum depression. So all this information together from our lab and from many other labs next suggested that mechanism implicating neurosteroid as neurosteroids biomarkers of postpartum depression and predicting the treatment or response of mood disorder and as well as PTSD, and that supplementation of allopregnanolone in allopregnanolone deficient subjects may provide a target pharmacological approach to treat mood disorders. Next, and in fact, this idea was then developed by Sage Therapeutics, by Stephen Paul, who is also a pioneer in the neurosteroid field, and together with Samantha Menzel-Brody, they studied the postpartum population given IV allopregnanolone, and so the results were mind-blowing because it was completely unexpected that allopregnanolone within hours induced a strong antidepressant effect. This IV allopregnanolone was given during 60 minutes, a 60-hour infusion, and so this after stopping this infusion, the antidepressant effect continued for a long time. Brexanolone was approved for the treatment of postpartum depression in 2019, but it's not anymore in the market because a new molecule next, the oral active allopregnanolone synthetic analog has replaced IV allopregnanolone. As we see here on the right side of this slide, zuranolone, which is the oral formulation of allopregnanolone given over 2 weeks, induces antidepressant effect after 3 days, and this antidepressant effect are maintaining up to 45 days, even when IV allopregnanolone, zuranolone is not anymore on board because the treatment only lasts 2 weeks. So this is really a very interesting discovery in the field because as we can see in the next slide, not only these results led to the approval of zuranolone for the treatment of postpartum depression under the name of Zurzuvae, but there is a clear advantage of these molecules over the SSRI. Next slide, in fact, shows that while individuals continue clicking that are given SSRIs, about 50% of them improve their symptoms. Individuals given allopregnanolone improve their symptoms with a much higher response rate. It's around 70%. The beneficial effects for SSRI are between 3 to 6 weeks versus 2 to 3 days. This in the treatment of postpartum depression is really very important because if a mother has postpartum depression with suicidal ideation, you cannot send her home with a baby. And it's very unfortunate because when that occurred, there were cases of suicidal and homicide. So this is really very sad. And so it's really very important to have a drug that is fast-acting, such as allopregnanolone, and can give the opportunity to send the mother and the baby back home. Also the relapse rate with SSRI is huge, and it varies in many studies, and instead using allopregnanolone is only 6%. So there is a really clear advantage in the field and the treatment of a psychiatric disorder, where we have a fast-acting molecule and a really short treatment course of only 2 weeks or hours, and then long-lasting antidepressant effects. Next slide. So now I will also cover some biomarker work that my lab has done in the field of epilepsy using neurosteroid-based biomarkers and treatment. Next slide. So in this case, we have focused on the relation between the allopregnanolone and sulfated allopregnanolone. As I mentioned to you before, the sulfated form inhibit the excitatory neurotransmission mediated by NMDA, and allopregnanolone potentiate GABAergic neurotransmission. Next slide. And so this was a study that we performed in collaboration with Marine Pharmaceutical. From this study, I hypothesized that an imbalance of inhibitory excitatory neurotransmission in subject with epilepsy, next, as it's very important and could be predicted by allopregnanolone sulfate levels in direction, as I mentioned to you, as a blocker of NMDA-mediated neurotransmission. So I also predicted that allopregnanolone levels could work as a biomarker to predict treatment response to the synthetic neurosteroid ganaxolone. So we found, the next slide, that lower level of allopregnanolone sulfate predicted the efficacy of ganaxolone in individuals where allopregnanolone sulfate levels were high. There was no response to the treatment of ganaxolone. So this is really very interesting because offer the very first predictive clinical biomarker in neuropsychiatric as in general and in the field of epilepsy. So this study, phase 2 clinical study, was run in my laboratory where we did all the biomarker characterization. And these results served in planning and designing next the phase 3 clinical trials, where patients were recruited for low or high allopregnanolone sulfate levels, and individuals with low allopregnanolone levels showed, so in other words, stratifying this epileptic population, it was possible to find positive effect of ganaxolone, so predict the efficacy of this treatment that led, next slide, to the approval of ganaxolone as Citalmi for the treatment of seizure associated with CDKL5 deficiency disorder, which is a rare disease, and it's characterized in children with epileptogenic seizures. And so this is really very interesting because next, this treatment was as an oral suspension was approved in the US, in the UK, in Europe, and also in China. And it's helping a lot of children around the world, which is really gratifying for our work. So what are we doing next? Next slide. And so there are a lot of neurosteroidogenic targets that can be exploited. So far, the only drug that have been approved for the treatment of mood disorder are those that activate indirectly GABA receptor, or that can be sulfated, and then they inhibit NMDA neurotransmission. However, there are a lot of other targets. I spoke before about the endocannabinoid crosstalk, and I will focus a little bit on this right now. Next slide shows that some years ago, I was really very intrigued by a paper that appeared in the literature showing that PA, or palmitoethanolamide, which is a food component, and it can also be produced in our body, by activating the transcription factor and nuclear receptor PPAR alpha in the spinal cord, was capable of inducing allopregnanolone biosynthesis and modulating pentobarbital-induced sedation, an effect that was prevented by PPAR alpha antagonist or by allopregnanolone biosynthetic enzyme blockers, such as finasteride. So given that PPAR alpha receptor are widely expressed in the brain in cortical limbic region, in a new study, I hypothesized that PA induced allopregnanolone levels in the brain could be really very important to improve anxiety and fear responses and depression in rodent stress model of PTSD. And so we did this study. Helena, do you want to close your microphone? There is some background. And so at the same time, there was also some evidence showing that PEA and the homologous OEA are decreased in PTSD patient in a manner that inversely correlate with the PTSD symptoms. So we ran this study in the animal model of PTSD. Next slide. And we found that allopregnanolone, PA was important for improving fear extinction in this animal model of PTSD, and most importantly, prevented the re-emergence of fear after passage of time. So in other words, facilitate also fear extinction retention, and also at the dose response in the ability of inhibiting anxiety-like behavior, and also a strong antidepressant effects. And all these behavioral effects of PA were blunted in animals that were knocked out. There were PPAR alpha knockout, showing that PPAR alpha was really very important in mediating the pharmacological effect of PEA. So how is this mechanism working? We found that PEA increased allopregnanolone levels, and also allopregnanolone sulfate in several cortical limbic structures, including the prefrontal cortex, and a lot more, like it was much more potent in hippocampus and amygdala, and also in the olfactory bulb. As you know, in the rodents, the olfactory system is really very important in modulating behavior. And so there was on one end the increase of allopregnanolone that potentiates GABA receptor, on one end the increase of allopregnanolone sulfate, which inhibits NMDA neurotransmission. Again, also in this case, as an underlying factor, we have an imbalance of excitatory inhibitory neurotransmission, and molecules such as PEA with a neurosteroidogenic feature are able to rebalance this deficit in neurotransmission. Next, so all these results were next. I already talked about all these things. Next. So as results of this basic research study, we were able to translate from the animal model these results into a depression population, and we are now running this clinical trial with the Sheba Medical Center and with Dr. Weiser, and this is a collaboration with another investigator at the University of Illinois, John Davis, and so this clinical trial funded by the Stanley Foundation as the goal of studying in a phase 2 exploratory add-on double-blind placebo-controlled 6-week trial the pharmacological effect of PEA. So we are crossing our fingers and hoping that these results will be positive. So finally, I want to point a little bit on next slide on PEA. As I mentioned previously, PEA can be found in food, in eggs, tomatoes, and beans, and peas, corn, and so on. And it can also be produced by our body. And it is really very interesting because these offer a lot of effects on developing and studying nutritional psychiatry based on these compounds. Next slide. And so several herbs that we use daily and even cooking, they all have micronutrients with the ability of affecting PPAR alpha function. And so this is really very interesting because these micronutrients, could be delivered to the brain where they can affect PPAR alpha in microglia. And PPAR alpha is also a very important anti-inflammatory target. And by activating PPAR alpha, we have an anti-inflammatory effect that could be also exerted by neurosteroids. But anyhow, by activating PPAR alpha, we have a start of production of a neurosteroid with pregnenolone that is taken up by neurons. And in neurons, pregnenolone is transformed in allopregnanolone that can activate GABA receptor that are inserted in the pyramidal neurons that produce allopregnanolone itself. And but this allopregnanolone can translocate back to the glia where it blocks the pro-inflammatory cascade mediated by toll-like receptor 4 and induce also an anti-inflammatory component. Allopregnanolone can also stimulate the BDNF expression and function. And so there are several mechanisms that are partially being studied, but there is so much more work that needs to be done in this field. And with the promise of developing new molecules that can be used for the treatment of neuropsychiatric disorders. Talking about nutritional psychiatry is also really very interesting. The PPAR alpha is highly expressed in the microbiome and where it is intercalated in the effect of diets. For example, high-fat diets promote inflammation, lipid accumulation, depression, and anxiety. Healthy diet, as we know, the Mediterranean diet, there are several clinical trials that have shown anti-inflammatory effect, reducing obesity and improving anxiety and depression. So my idea and one of the hypotheses that we are running right now is that activation of PPAR alpha in the microbiome by healthy diets or diets with a high content of PEA may stimulate the bacterial population that produce short-chain fatty acids. These short-chain fatty acids have the ability of activating PPAR alpha, blocking the pro-inflammatory cascade mediated by toll-like receptors, and instead stimulating microbiome production of allopregnanolone that can function as a mediator of gut-brain axis. And so the allopregnanolone produced in the microbiome can translocate to the brain and induce all the beneficial anti-inflammatory and behavioral effect that we know. So this is really very interesting because next, thank you. We are studying a lot of this mechanism with my collaborators here in Brazil, for example, with Leandro Agacci, who was a PhD student with Cristina Velar, and he also did part of his PhD in my lab in Chicago. So now we are collaborating to discover molecule that can be effective in the treatment of stress-related disorder following this mechanism that we are discovering. We are also studying next with the Universidade Federal do Paraná in Curitiba with Professor Cristina Stern, the effect of cannabidiol, working on PPAR gamma receptor, and trying to understand better the mechanism of this molecule and the mechanism involved in the effect of cannabidiol in animal model of PTSD and as a facilitator of Phe extinction. And I'm also very excited about next a new grant that we have just received in this is the universal grant. We received a notification of award 1 week ago, and this is a collaboration with Universidade Federal de Porto Alegre. Our coordinator will be Professor Helena Maria Barros, and we have several other collaborators, including Professor Gilda Neves and Isis Souza from Universidade Federal de Rio de Janeiro, as well as Mauricio Schulernin and Felipe Borges Almeida, who are really two promising young scientists that worked as a PhD students in the lab of Helena, and they were also in my lab in Chicago. So we are very excited about this study that promise of developing neurosteroid-based biomarker and studying the intranasal pharmacological effect of allopregnanolone in a preclinical model of neuropsychiatric disorders. And thank you next again for the Helena for hosting me here today and for our long-lasting collaboration, which is really always very stimulating, and it's a really very important reason for me also to come back every time here to Brazil and keeping all this collaboration alive in several universities in Brazil. So with this, I really thank you very much all for your attention, and thank you for having me here again. It's an honor as usual, and I want to thank also my team of very hardworking young scientists that have contributed to all these results, and also many more that I couldn't show today. I can promise to show in the future. And in particular, I want to mention the Brazilian Mauricio Schulernin, Felipe Almeida, and Leandro Agacci, but also Elias Cruz, who is Argentinian, and there is Carlos Sánchez Barbosa, the Spanish, Francesco Matriciano, and many other talented individuals that made all this possible. I also want to thank the CARE Epigenetic Studies in Alcohol Use Disorder and the UIC Center for Depression for having me, as well as a number of agencies that have supported my research over the years, including the NIH, Veteran Affairs, Department of Defense, and so on. Thank you very much for your attention.
Thank you, Graziano, for this very interesting and inspiring talk for us today, so we can think of many other projects to try to run with you, and having other Brazilian young scientists joining the efforts to understand how the neurosteroids are interacting with the brain, or the brain is interacting with the neurosteroids, and how we can improve psychiatric treatment for women and for men during all those development phases that we have in. So now the idea for us is to have to see the questions that we have from our public today, and then thinking of how to interact during this following session of questions.
So there is a question here, and I can read it and provide an answer from Janaina Menezes Zanovelli. Probably she's online, but anyhow, one of the biggest problems with drugs that act on the GABA receptor is dependence. What do studies show about brexanolone used for postpartum depression and other PAM compounds? PAM is positive allosteric modulator compounds in relation to the development of dependence. Has this issue been a concern at the time of increasing medicalization? Okay, this is really very interesting question. So here, probably Janina was referring mostly on benzodiazepine and all the effects that benzodiazepine gives in terms of tolerance and dependence. So benzodiazepine, a positive allosteric modulator, as neurosteroid are on GABA receptor, but there is a huge difference. While benzodiazepine act mainly on a synaptic GABA receptor, neurosteroid work mainly on extrasynaptic GABA receptor. So this is a huge difference because, for example, benzodiazepine don't act at GABA receptor formulation, including alpha-4, alpha-6, and delta subunit that are mainly the composition of extrasynaptic GABA receptors. So given this difference in mechanism of activating GABA receptors, we don't have with neurosteroid all the dependence and tolerance liability that we find with benzodiazepine. So while there are no studies so far that have studied the brexanolone dependence, we already know from basic research studies that this is not the case when we treat animal or humans with neurosteroid-based treatments. And also there is a difference. We know that benzodiazepine induce sedation, and instead for neurosteroids, the anxiolytic antidepressant doses are devoid of sedative properties. So there's a big difference in terms of mechanism that reflects into pharmacology. Also, thanks for the brilliant. Okay, so let's see if there are more
questions here by Carla, where she wants to understand a little bit about the feeding behavior of the animals that are isolated, regarding the idea of having, some foods that may improve their behaviors. So what have you done about that, or what are you thinking about that?
Okay, so this is also a really great question, and we had thought about doing something in this concern because we are, and I was talking with my postdoc about this even 2 days ago. And so we are really very interested in studying the effect of diets in this animal model of stress-induced behavioral dysfunction by using a functional diet that can mimic a Mediterranean diet and contrast that with an obesogenic diet. So we believe that the obesogenic diet, by decreasing PPAR alpha expression, is able to give also a number of behavioral deficits, including anxiety and depression. As opposed to the Mediterranean diet, they should be rich in micronutrients that are PPAR alpha activators and should improve emotional behavior by also increasing allopregnanolone. So this is really very interesting, and there are a lot of implications. One of the things that I didn't discuss today was the effect of alcohol. We have grants to study alcohol use disorder from animal model to the clinic. And one of these granted basic research study that we are performing is the effect of alcohol on the expression and epigenetic reprogramming of PPAR alpha. And I mentioned to my postdoc that I see alcohol as a bad diet, just as an obesogenic diet, that has the ability of decreasing PPAR alpha expression and inducing a number of effects, from a pro-inflammatory effect to effect, the deleterious effect on behavior, and so on. So this question is really very interesting, and it's in plan to study the effect of diets on this animal model, and is definitely very informative to improve diets. And in the past, I also have been involved in grants in Chicago that were looking into improving diets for children at school and introducing functional diets, and not only to improve behavior of people, but also to improve the knowledge and educating people to eat well. Because as you know, in the United States, there is a huge problem with obesity and inflammation. Obesity and emotional behavioral dysfunction are all factors and that are all linked to each other. So it's possible that in the future we will have really important discovery that can take care of all these deficits.
And also, Graziano, if we may, we are, finishing our time here today, but there's a very interesting question regarding the use of neurosteroids in menopause. Since we know that over there, or during this time, we also might have very important changes in progesterone synthesis and other metabolites of the neurosteroids. What do you think about that? What you have done regarding that kind of research?
Okay, so this is also a really critical point and question. Unfortunately, menopausal women are being left behind. One of the goals of the past First Lady, Jill Biden, in the United States was to take care of women during menopause and stimulate studies in postmenopausal women. And because the state of the science is so backwards, these women are basically forgotten. And so it's really very important to provide means that can help these women, go through this critical period of menopause and the postmenopausal period. And it's really very important to provide pharmacological means for people in need. So I collaborate with Pauline Mackey, who is an expert on postmenopausal women, and this is also the leader of this training grant that we just received to educate, especially women, not educate, train them in the field of women mental studies. And so what we do, we want to understand better what's going on in by studying the blood in postmenopausal women, and how can we better assist them during this postmenopausal period. So far, we are now publishing a paper also here in Brazil with Leandro Agacci and Science Valley that has studied the subcutaneous insertion of estradiol in postmenopausal women to improve symptoms that arise during the postmenopausal time. But we still didn't do any study related to studying neurosteroid and how neurosteroid can benefit these women during this critical time.
So thank you so much for being here today with us, and for all, the long-term collaboration that we have with so many universities and industries, pharmaceutical industries and researchers here in Brazil. We hope that we are going to be, studying more about this research, this subject. And again, I thank you so much for the talk that we heard today.
Yeah, thank you so much. I really treasure our collaborations, and there is so much that we still need to do. So we need a lot of young, enthusiastic, and passionate scientists that can join these teams and that can progress the future of neuropsychiatry and neurosteroid-based studies. So I hope that these talks are also important to stimulate them and invite them to join the neurosteroid teams for the future.
Thank
you
you
very much again.
Thank you, Helena. Thank you, Graziano, for the excellent session. Helena, if you can please share the slides again so I can conclude the session. So I'll change to Portuguese. Bom, muito obrigada a todos os participantes pela sessão de hoje. Como só relembrando, todas as nossas apresentações, a rede RNP depois elas nos disponibiliza a gravação da sessão, e esses vídeos ficam disponibilizados no canal do YouTube da SBEFT, que está aí no endereço, né, colocado aí no slide. E esse sítio do YouTube da Sociedade Brasileira de Farmacologia e Terapêutica Experimental, a SBEFT, que nos apoia nessa iniciativa, ele tem uma coletânea, nós temos um portfólio que vem já de todos esses anos que temos tido essas atividades. Então, essa em alguns dias será um vídeo que será colocado lá e fica livre para vocês que estão aqui na apresentação, revisitar a apresentação, e qualquer outro colega. Então pedimos que ajudem a divulgar, né, para que nós possamos ter então esse material educacional auxiliando, né, dentro da universidade e para o público em geral. Gostaria de colocar aqui no próximo slide, por favor. Mais uma vez lembrar a participação de vocês, no anterior, Helena, por favor. Lembrar a participação de vocês, aqueles que necessitam ou querem um certificado de online, lembrando que o registro da presença é válido apenas durante a apresentação aqui, de 1h30. E no próximo, por favor, a próxima agenda, então, em outubro, nós teremos o tema sinalização não canônica de proteína G como alvo para o desenvolvimento de novos medicamentos, e que será apresentado pelo nosso colega Lucas Tabajara Silva, que é docente e pesquisador da USP em Ribeirão Preto. E a sessão será moderada pela professora Roseli Godinho, da UFCSPA, Escola Paulista de Medicina. Essa sessão será no dia 15 de outubro, um pouquinho antes de todos nós estarmos nos reencontrando presencialmente aqui na cidade de São Paulo, na oportunidade do congresso anual da SBEFT, né? Que vai ser entre 21 e 24 de outubro. No próximo slide. Eu gostaria de finalizar novamente agradecendo a toda a audiência. I would like to conclude again. Thank you so much, Graziano, for being here. Thank you, Helena. And just to conclude, so this is one more session of our SIG Farmaco. E então nos encontramos no mês que vem. Obrigada a todos. Thank you.
Thank you.
Emiliano, can you
stop the recording?
Thank you.